soybean for biofuel, for motor oil, for t-shirts, for ink, for crayons, for candles, all sorts of really cool stuff can be made out of soybean. Soybeans long been considered cold intolerant. This is a plant before it went into the cold. Nice green leaves, super happy. This is 2 weeks after the cold. Leaves are drooping, very, very yellow, some tissue damage occurring. So the question overarching for this work is, why is soybean cold intolerant? Why doesn't it respond like other plants? Soybean is a crop, which means it's been domesticated, and it's been domesticated over a long period of time. So we have selected certain traits. We wondered, if we're selecting these traits, could we be selecting against cold tolerance, right? Could other traits have been lost? So one of the things that I looked at was comparing the domesticated, this is glycine max, versus the undomesticated, which is the closest to a wild soybean that still exists on the planet. And this is glycine soda. You can see just by looking at them, right? Glycine max, really, really big, huge leaves. These are really viney and tiny, and their seeds look exactly that same way. Soybean seeds are nice and big. Soja seeds are really small and difficult to work with. A lot more seeds come out of here, lot less seeds. So in this process, could we have interrupted the cold tolerance? One way that we can measure cold tolerance, it's called an electrolyte leakage assay. So in this assay, you're just taking leaves, some of which have been previously exposed to cold temperature, some of which had not. You're cutting them out, and then you're putting them in test tubes and exposing them to freezing conditions. And then you take different time points, and you can measure the freezing tolerance based on how much conductivity is in that water. And that conductivity is coming from damage to the cell membrane. So when the plants are freezing, it can pop the cell membrane. The electrolytes that are inside the cytosol leak out into your DI water, thus increasing its conductivity for electricity. One of the things that was really interesting when we first started this project, so here's our ion leakage. It's done by percent, so how much leaked when I first measured it, and then you freeze them really, really hard so they're all broken. So you can compare it to how many cells total breakage would look like. And then several different temperatures. Your dashed and open symbols are those that were held at 22 degrees prior to this assay, whereas your dark and closed symbols are those that were exposed to 4 degrees for 2 days. And the light leakage percentage is a lot less for these ones that have acclimated, which was a really interesting observation considering this plant has been cold intolerant classified. It is responding, right? Cold acclimation is occurring. It's just more moderate than Arabidopsis. For example, down here we've got -1 to -3. With Arabidopsis, that would be -6 to -9 is where you would see similar. So it is responding just in a more moderate way. We wanted to know, okay, cool, it's responding. Does this response increase over time? And remember, this is electrolyte leakage, so you want less leakage, so like golf, low score wins. You don't want it to be really high leakage. And this is the number of days that they spent at 4 degrees prior to being put in the assay. And 0, right, where there was no cold acclimation, you have like 95% leakage. They're really, really stressed plants. Then 1 day drops, 3 day drops, 5 day, 7 day, 10 day. So they are capable of cold acclimation. It takes about 5 days for 50% of the cells to be okay versus 50% of the cells to be damaged. So that was a nice little benchmark for us. Okay, it takes 5 days, 7 days is also good, so that we could set up our next experiments to use those timings. Soybean are classified based on their maturity group, and while this was based on photoperiod, right, how much light this area gets, one of the things you might notice is this is also based on north-south. So 000s, they're up in Canada, right? And then you have all the way down to Florida, soybeans can be grown. So this also led to the idea of perhaps not only are there different cold tolerances, perhaps it was this selection that also helped with that. So would these northern varieties be more cold tolerant than southern varieties? And would this hold true and hopefully be better with our undomesticated glycine soja? And that's exactly what I looked at. So here we have ion leakage, and these are just different types of soybeans. So they're called cultivars, and we have from a 000 all the way to 7 of glycine max, and then a 0 to a 4 for glycine soja. And we're comparing the non-acclimated in the dark, those that were held at 22 degrees prior to this assay, versus the ones that were held for a week at 4 degrees C prior to this assay. And in every single case, we see acclimation, right? These clear bars are lower than the dark bars. And when we look at the northern versus southern, here's our northern variety, and then here's our southern variety. So there is a modest increase, right? When you are a 000, after these same conditions, you got about 22% leakage, whereas Harbor's got almost 50. So there is a slight difference here. However, when we look at glycine, it's pretty stable. This dampened our enthusiasm for would there be cold-tolerant genes present in this undomesticated population. When what we're seeing is no matter where they're coming from glycine soja, they have about the same leakage, and it's all within the same range that glycine max has. So there's probably not good genes here. Okay, well now we got to go back to the drawing board. So we've thought back to, okay, what do we know about cold tolerance? What is the molecular control of this? And for that, we go to Arabidopsis. Again, Arabidopsis is a cold-tolerant plant, and this is how the pathway works in Arabidopsis. This pathway is called the CBF cold-responsive pathway. If this primary transcription factor, CBF, is knocked out, the plants cannot acclimate and cannot survive the cold. So it controls 15% of the genes, but they're 15 critical percent, because without it, it cannot survive. When you look at Arabidopsis, exposure to cold increases cytosolic calcium levels. This activates a kinase cascade, and then SEAS1 simulates ICE1 to prevent its degradation by HOS1. OST phosphorylates ICE1 also to prevent its ubiquitination by HOS1. And then ICE1 is a transcription factor which activates our CBF DREBs. From there, CBF DREB go to activate CRT DRE containing elements. So this is just the consensus sequence that it seeks out, and it's found in most of the cold responsive genes in Arabidopsis. And then you get the process of cold acclimation. So we know all this is true in Arabidopsis, but what about soybean? Is the fact that soybean's cold intolerance potentially due to they don't have homologs to these genes? Are these genes just not present and or not working? And Dr. Yamasaki looked at this a couple years ago, starting with the dihydrins. So this is a family of cold responsive genes that's very important. When you lose dihydrins in Arabidopsis, you lose cold tolerance. So it was a good place to look for homologs in soybean. And he found homologs in soybean, but they did not increase, the transcript levels did not increase in the cold. So these are not increasing in the cold. So at least from here down, this pathway is definitely not working, even though the genes are present. So he took a step up the pathway and did a search for homologs to the CBF DREBs, so from Arabidopsis in soybean. And he found 7. GM stands for glycine max, and AT stands for Arabidopsis thaliana. So there are 4 Arabidopsis CBF DREBs, and then there were 4 soybean CBF DREBs that were the closest homology to Arabidopsis. It was those 4 that the lab kind of moved forward with to see, okay, they're there, are they activated appropriately? If they're cold responsive genes, we'd expect to see the transcripts increase just like you see in Arabidopsis. And that was the next step. In soybean, when you measure the transcripts of DREB1A and DREB1A2, and this is 2 different biological replications, and you're looking at leaves and stems, right? So 2 different organ tissues to see what's happening. The fold changes were massive in the early cold stress. So here, 1,000-fold, 400-fold, 450-fold, right? There was a large, massive increase in these transcripts. If you were to look at Arabidopsis transcript levels, this is exactly what it looks like. There's a really sharp increase in early cold stress, and then it comes down and maintains a slightly higher basal level while it's in the cold, but there's only a short burst. So this looks exactly like Arabidopsis. Okay, we're still not getting cold acclimation. We're still not getting increases in these CRT-DRE genes. So what's going on here? Well, next hypothesis, are these incapable of binding to CRT-DRE promoters? Perhaps over evolutionary time, they've just mutated where their active site no longer is able to bind to the right targets. So to answer that question, Dr. Yamasaki took 3 of these and he put them in Arabidopsis under constitutive expression. So they were on all the time. And before we get too far into that, I want to show you what Arabidopsis looks like. So this is wild type Arabidopsis, 0 hour, no cold. Wild type Arabidopsis, 24-hour after cold. And these are 3 genes that contain those CRT-DRE elements that CBF binds to. You can see all 3 are increased in Arabidopsis in the cold. And when you take those soybean CBF drebs, hook them to a constitutive expression promoter, and you look at these same transcripts without any cold exposure, those same transcripts increase, or fashion. So this suggests that, yeah, they are capable, at least in a functional cold system, the presence of these genes are enough to activate the downstream targets. And when you do freezing tolerance, they act like they're acclimated plants, even though they've never seen cold, right? You just take them and put them right in the freezer, and they respond just like an acclimated plant would. So right now, we know that soybeans are transcriptionally cold responsive in soybean. They go up really sharply and come down like they do in Arabidopsis. And when you put them in Arabidopsis, which is a functional cold tolerant system, they activate those downstream targets. So why is soybean cold intolerant? Could there be something outside of this pathway that's influencing it, that's making it maybe inhibited so that it can't respond quite exactly the way it should? Back to the literature, where I was able to locate IN3. So this is IN3 and its homolog EIL1. When they're frozen without any exposure to cold, you see a lot of death, right? They don't acclimate well. And when you look at transcripts, here's the CBF proteins, then here's those downstream targets. At 22, so this is at normal temperature, in these knockout plants, all of these transcripts are upregulated, suggesting that this does have a negative control. And this paper went on to show that, yes, IN3 binds to the CRT, to its motif in CBF. So at least in Arabidopsis, this is happening. But we don't know much about soybean. Before we dive into soybean, just want to refresh everybody about ethylene. Ethylene is a very small molecule, but it regulates lots of stuff. Growth, ripening, senescence, abscission of the leaves. And you can look here, right? You go from green to banana bread thanks to ethylene. And we know that it's involved in the stress response. So we want to look at it a little closer. When you look at the ethylene signaling pathway, you have ethylene, its receptor, a kinase, a stabilizer, a ubiquitin, E3 ligase, and a couple of transcription factors. This pathway is really cool to see in motion. So instead of stepping you through all this, we're going to look at it in a little bit of a cartoon fashion. In the absence of ethylene, so notice there's no ethylene over here, the ethylene receptor is keeping CTR1 activated. So it's phosphorylating IN2. This is a kinase, and it's phosphorylating IN2. Inside the nucleus, IN3 is being marked for degradation by EBF1-2, so it's degraded by the 26S proteasome. When ethylene shows up, it binds to its receptor. CTR1 is deactivated. This allows self-proteolytic cleavage of the C-terminal from IN2, and it moves into the nucleus where it stabilizes IN3 and disrupts this ubiquitination process so that it's no longer broken down by the proteasome. Now IN3 can bind to its targets, and then there's a couple more ethylene responsive transcription factors, and then the broad suite of genes can be turned on. So why are we looking at all this, right? I told you I'd talk about cold, and here I am talking about the ethylene pathway. Well, remember, IN3 has also been shown to negatively regulate our CBF-DREB cold pathway. So now let's look at it in soybean. These are the different homologs in soybean to those same genes I just showed you from the ethylene pathway. So we have CTR1, ETR1, IN2, IN3, EBF1. And when you look at the IN3 family, these are transcript folds, 1 versus 0, 24 versus 0, and 24 versus 1 in Arabidopsis out here. And notice there's very little color change. Now we're going to compare those same times in these soybean homologs. And they are upregulating based on the darkness of our scale, right? If we look at this heat map, they're actually increasing, where they're not doing anything in Arabidopsis. The positive regulator, IN2, it goes up a little and comes down, but it goes up a lot in soybean during the cold. And those negative regulators at the very early cold stress, they go down, where in Arabidopsis, they go up a little bit. So even though this is a post-translational control of IN3, more transcripts could mean more of those products, which then is going to shift the equilibrium one way or the other from repression versus stimulation. That led me to the question, okay, well then is ethylene negatively regulating soybean? Because obviously these genes are not expressed the same way they are in Arabidopsis. There is a difference here. So is this difference important? Ooh, wrong direction. So if I block ethylene, you should increase IN3 and decrease CBF-DREB and cold tolerance. And that became my first step. To do this, I generated a tool. This tool is a cold reporter line in soybean. So I have 3 independent transformation homozygous lines of soybean that I can use. They have the entire RD29A promoter in them. And most of the time when you see this in the literature, they only use this portion with these 3 CRT DREs. And sometimes they put like 3 or 4 more of those in there just to make it super responsive. This is the native Arabidopsis promoter. And it's driving GFP GUS. This is matched with an herbicide resistance gene so that I could quickly screen these lines. If they had that resistance gene and then also they had the construct, you'd get very little tissue damage. If they didn't have that gene, you get quite a bit of tissue damage. So it was a nice screen to be able to quickly go through these lines and identify our 3 homozygous. When we use GUS, we use the fluorometric assay. So there's 2 ways you can do GUS, and we like the fluorometric assay. In this case, GUS is an enzyme. It breaks down complicated carbohydrates. So if you feed it a specific carbohydrate, MUG, it cleaves one of the rings off, and that ring is now fluorescent. Right? So you have a fluorescent particle. The more fluorescence, the higher your relative fluorescent unit, and this slope is then correlated with the activity. So I'm going to be showing you values that are based on the slopes of the fluorescence. Whenever I talk about GUS activity, it's looking at that slope. When you look at GUS activity in the 3 reporter lines, here's our non-acclimated, so 22 degrees, water treated with silver. Hey, look, our downstream target is going up even in the absence of cold. 4 degrees, and then water sprayed, and then silver sprayed. And once again, we see an increase in GUS activity. So at least at this point, it looks like I might be on the right track. So when you do silver nitrate and you spray the plants, you do get an increase in GUS activity, even though we're turning off ethylene. Well, the ethylene signaling pathway is super complicated, so you can't just do one thing and say, look, I fixed it, go me. So we did silver first. There's also more specific inhibitors. For example, 1-MCP, it has the same mode of action as silver, so it blocks the receptor. We also did 2 stimulators of ethylene to see if this also goes the other direction. One of them is ethafon. Ethafon is quickly metabolized by the plant into ethylene. So it's like feeding it ethylene even though you're not using the ethylene molecule itself. Nice thing about ethafon is it's powder, so you can make up your solutions. Ethylene's a gas. ACC is the biological precursor to ethylene. right? So you end up with one step up the metabolic chain. You give it a lot of ACC, the plant will turn that into ethylene. And then the last inhibitor we used is called AVG, and it blocks the synthesis of ACC. So if you're blocking the synthesis of ACC, that plant can no longer make ethylene. So in this case, we're blocking and stimulating the biosynthesis pathway of ethylene, and then we're also blocking the receptor, or the ability to sense ethylene. So this lets us come at the problem in several different ways. Did the exact same thing looking at Gus. This time I picked a single reporter line since all of them seem to respond similarly. And we're looking at, cold responsive gene. Here's 22. Again, we've got some changes, but this is where it gets really cool. All three of our inhibitors have an increase in Gus activity when exposed to the cold. So just by blocking ethylene synthesis and blocking its receptor, we get an increase in Gus activity. Awesome. What's going on behind this in the plant? So one of the things to do is, okay, well, let's actually measure the transcript levels of IN3 and actually measure the transcript levels of CBF and compare them. These are exactly what I did. Relative transcript levels. Now we're measuring IN3, and again, orange is going to be inhibition, green is going to be stimulation. And when you stimulate the ethylene pathway, you get an increase in IN3 transcripts. When you inhibit it, you see a decrease. So at least at the IN3 level, this is working the way I expect it to. When you go down and check the level of CBF transcripts, you get an increase in CBF transcripts with inhibition and a decrease with stimulation. Now, one of the interesting things that fell out, of course, is this giant silver bar that I'm sure nobody can take their eyes off of, right? Like that's a huge increase in Gus activity just by treating with silver nitrate at 22 degrees. And we think this is due to some off-target impacts of silver. So that's one of the reasons why you use other ethylene inhibitors, because silver not only messes with ethylene, it messes with a lot of other things in the plant. So you want to make sure that what you're seeing is ethylene. Oh, yeah, and that goes up. So the next step was to go down and look at some targets of CBF. These blue triangles that are in the yellow circles, that's the CRT-DRE element that CBF binds to. So we would expect if CBF transcripts are going up, hopefully the protein's going up, CBF is binding, creating more. If it's going down, we should go the other way. And all three of these genes, they were cold responsive. So CBF is binding to them. They are being increased like you'd expect. And then when you stimulate ethylene and CBF transcripts are down, there's a pretty good decrease in transcript level for these genes. However, when you inhibit ethylene, there's no universal response. So even if you increase CBF transcript levels, you don't seem to get an increase in at least these downstream genes. And as one of our controls, we also looked at a gene that does not have that CRT-DRE promoter, and it was also not regulated in the cold. So at least we know that genes that should be regulated are, and genes that shouldn't aren't. Well, even though we didn't see a whole bunch of cold responsive genes, still took a look at cold acclimation. I only looked at some of the genes. You can't look at all of them without doing a big RNA-seq experiment. So in this case, we're using electrolyte leakage, which we looked at before. And with electrolyte leakage, the treatment of ACC increases the leakage, which means the plant is less cold tolerant, right? This is inverse. So when you're stimulating ethylene, it's less cold tolerant. And when you treat it with silver, when you're inhibiting ethylene, it's more cold tolerant. But the other inhibitor and stimulators didn't have much of an effect. So again, it's not universal. We also looked at MDA content, which is a measure of how much stress, oxidative stress, is on lipids, so how many lipids are being oxidized. And when you look at both silver and MCP, they actually go opposite directions. So once again, not really sure. We're not seeing a nice ending. What I did do was I also looked at photosynthesis. So I'm a huge photosynthesis buff. This is my favorite biological pathway on the entire planet. What you're looking at here is called FV over FM. It's basically the efficiency of photosynthesis. So it ranges from 0 to 1. 1 would be 100%. Every single photon that's being captured by the sun is going right into photosystems and being made for energy in the plant. 0 would be 0, right? So you don't usually see that because your plant is dead. Typically, values are between 0.7 and 0.8 for happy, well-treated plants. And in our controls, that's what we saw, a little over 0.7. However, when you put it in the cold, you get a loss. And when you treat it with any of the stimulators, you get more depression. So the photosystems are pretty taxed just by the cold. Because if you manipulate them at all, they go down even more. What's driving this? So this is just an efficiency, right? Like just looking at everything and saying, okay, it's this much is going towards photosynthesis. Well, the really cool thing is you can parse out some of this stuff using different methods. So I looked at how many photons are actually trapped by photosystem 2. So a photon hits the reaction center. It's either going to be trapped and sent to the quinone, or it's going to be reflected as fluorescence, which is what you actually measure here, or it's going to be dissipated as heat, right? So there's 3 fates. So in this case, I'm looking at how many of these are actually trapped by that photosystem. And in the cold, less photons are being trapped by photosystem 2, and you get even more decrease. Okay, well, if they're being trapped, where are they going? Looked at how many photons are being dissipated away from PS2. So this would be through non-photochemical quenching, right? So it's not the part coming out in fluorescence, but it's the part coming out the plant's releasing as heat or with xanthophylls. And it increases in the cold, and then increases substantially more with ethylene. So all of this tells us that, at least at a photosynthetic level, cold stress is very hard for soybean, right? It's at its bottom edge. And then you start to see it crash out after that. And this crash is coming from less photons being trapped and more photons being shuttled away. What I don't know is exactly why that's happening, but that's another story for another day. So let's take a quick review of what we've seen so far. Again, if we're seeing something in green, that means ethylene pathways being stimulated. Orange means ethylene pathways being inhibited. And then cold is just that general cold response without changing ethylene at all, what naturally happens in the cold. And when we looked at the GM ion 3, in the cold they go up, just like DREB1 goes up. However, when we stimulate ethylene, they go up even more. When we inhibit ethylene, they go down even more. And then for DREB1, when we inhibit ethylene, they go down. And when we stimulate ethylene, they go up. So at least at this level, it does seem that GM-IN3 is suppressing the transcripts, right? It is affecting the transcripts of CBF. But with our cold responsive genes, in the cold they go up, with inhibition they go down, but they don't change when DREB1's going up. So that then leads to not very much change going on our actual reports of cold tolerance using our techniques for physiological stress that we look at. Conclusions from this part, right? Soybean, it's cold tolerant. Yay! Just not as much as other plants. But at least we know it's there. The possibility is there. Unfortunately, the wild or the undomesticated glycine soja didn't seem to have a lot left in it, right? There wasn't any genes that said, any response that said, ooh, we selected against cold tolerance, because they had the same cold tolerance as the domesticated. So that dampened the enthusiasm. When we looked at ethylene signaling, we saw that it negatively impacts CBF, DREB, and soybean. And we suggest that this is because IN3 is binding to its consensus sequence here in the promoter of DREB1A and blocking the transcription. If you remember way back from the cold tolerant pathway, ICE1 was the transcription factor that I said induces CBF expression. It's actually named ICE1 because it induces CBF expression. Like that's its whole name. And the ICE1 consensus sequence, at least for DREB1A, is very close to the IN3 consensus sequence. So you have some IN3s binding here. Well, you know nothing else is going to be able to get on that promoter. So that's at least one of our theories. Cold tolerance, in my opinion, we need to go downstream of CBF, at least at the transcriptional level. For example, we'll go back to this, right? When IN3 is going down, CBF is going up. When IN3 is going up, CBF is going down. When CBF is down, its cold responsive genes are down. So upregulating CBF transcripts doesn't do anything to those cold responsive genes. It doesn't do anything to the cold acclimation. So whatever's happening in soybean that's keeping it from reaching its full cold tolerance potential is going to be somewhere between the transcription and the activation of the cold responsive genes. Right, so we've narrowed it down to that field. I've actually got a couple ideas of what we should do and where we should go from here. So back to our big pathway. We know that everything up here seems to be fine because our CBF transcripts go up, they go down, but the downstream targets don't necessarily do the same. So let's look at things that affect the CBF protein. 1433, for example, will phosphorylate CBF DREB1 and make it more susceptible to degradation, right? So it makes the protein go away faster. We already know that protein's turned over fairly quickly, but in the presence of phosphorylated 1433, that turnover rate is increased. So perhaps this protein is misregulated in soybean and it's not being turned off the way it is in Arabidopsis. So even if you're increasing the transcripts, if you have something that's marking your protein for degradation, you're not going to get a lot of downstream effects. On the other side, OST1 actually phosphorylates BTF3, which is a subunit of the nascent assembly polypeptide complex. And it's shown if you overexpress BTF3, the half-life of CBF protein increases dramatically. So this is, at least in Arabidopsis, is stabilizing, again, the CBF protein. Potentially in soybean, maybe this connection, BTF3, is not getting activated in an appropriate manner to be able to stabilize the protein. So if you increase the transcript and you increase the protein, it's still not being stabilized, you're still not getting anything downstream. And the last that I have as an idea, there's lots of other possibilities. The last of my ideas is called mediator 16, or MED16. It's a subunit of the mediator complex, which is critical to RNA polymerase and transcription. And it has been shown in Arabidopsis that the loss of MED16, CBF DREB1 is not able to bind to its promoter elements. That MED16 is critical in the recruitment of CBF to those promoters, critical to the recruitment of RNA polymerase to those promoters for those genes to be transcribed. If this gene was inactivated or somehow non-functional in soybean, we'd see the responses we're seeing, right? We wouldn't get massive upregulators of CRT-DRE because it's required for at least a little bit of MED16 to be around. So those are my three potentials, right? Perhaps it's being inhibited too much. Perhaps it's not being stimulated. Perhaps it just can't bind to the promoter. And so you're not getting transcripts. The protein levels could be fine. So the next steps really need to focus on the protein level of CBF and the transcription level of those cold responsive genes to see if we can narrow this down at all. Science itself is never performed in a vacuum, right? There's always people that are along the way. And the most important person along this way is my son, Wayne, who is back there. He has been my most trusted lab assistant. He comes in, he racks pipette tips for me. He waters soybeans. He learned how to pipette. He was all in a lot of times. And, you know, 1st through 5th grade, he pretty much spent a lot of his playtime in my lab. So he's been very patient and helpful, and I would not have been successful without him. Additionally, my parents, who are also in the room, they provided so much behind the scenes work y'all will never even understand. right? Like, I'm a single mom. I have a kid. He gets sick. He has breaks from school, all this. I go away to conferences, right? All this stuff. And they step in. They take him to their house. They step in. They're always there. If I have any kind of problem, they're right there if I need help. And then my grandparents, who are also in the room, especially my grandpa, who was Professor Robinson at Manchester College for a very, very long time. So he pretty much put me on this path, right? Like, I grew up with him saying, hey, I teach science teachers how to teach kids. Come here, guinea pig, right? And so then I'm like, science is awesome! And now I want to be a professor, but teaching biology instead of education. Though I still do have a pretty good love of education. You have so many friends that keep you from completely losing your mind. We've got Sky, who's my best friend. She's not here, unfortunately. Melissa actually learned how to do RNA extraction so she could help me with my gigantic experiment. So she came in, never done science. She's like, I can do this. So I taught her. She did like 80 RNA extractions for me. It was fabulous. That's why Kent's in the parentheses, because all he did was say she could come down. My friends Sarah and Melanie, they listen to me when I'm like, why did I do this? What part of me thought this was a good idea? And I have a lot of people on Twitter who have sent really good vibes for me and have stepped in and said, hey, I'll edit for you. I'll do whatever you need. Just let us know how we can support you. So it's been very, very helpful. And of course, local, the department, my committee members, John and Brenda and Lotta, they've made so many helpful comments to say, you know what, why don't you do this? Or you might be just chasing something you're never going to find. Maybe we should dial this back. All my Randall Lab members, I can't think of a single one that hasn't been fun to work with. We have a lot of laughs. He takes us to the state fair, so that's always fun, right? Like free food on the advisor. The CTL, probably the only reason I have my job right now is because of all the training they provide to help those of us who want to really teach at small colleges get on our feet to be able to do that. Department of Biology, y'all know, in the hallways, you listen to each other, we complain, we cry on each other's shoulders. You get all the support from Angie and the rest of the staff. Plus, they paid my salary, so thank you for that. The United Soybean Board provided a little bit of money for helping with the RNA-seq and the transgenic soybean lines. And last but certainly not least, Dr. Stephen Randall for taking a chance on a 30-plus-year-old single mom who really, really wanted a PhD. Not a very traditional student, came with baggage, and he said, yeah, let's do this. And here we are. We have done this. So with that, I will take any questions anyone has.